You're listening to the Common Descent Podcast. Hello, Will.
Hello, David.
Hello, everybody, and welcome to Common Descent, a podcast about paleontology, evolution, and the history of life on Earth. This is episode 210. This episode is about bones. That's right. 8 years and 200 episodes into our podcast about paleontology, we decided that it's high time we talk about bones.
I'm sure a lot of you have been very confused for the last 200 and some odd episodes.
People are going to finally know what we're talking about.
We are usually so good at defining terms. We've skipped this one majorly.
You keep using this word and you just never, I don't know. This episode, we are going to do an in-depth exploration of bones. We will talk about what bones are, how they work. We'll get into some of the evolutionary history of bones, and we will talk about some of the ways that bones are useful for paleontologists, and we'll get into some specific methods and techniques that paleontologists can use to get as much information as possible out of the bones of ancient animals.
This is one of my favorite kind of topics we get to do, where we get to dive into a seemingly mundane topic, and it turns out to just be so cool.
This is another one of those that, if we do it right, this will change the listening experience for almost every other episode of this podcast.
Yes.
So this is how we get people to keep listening to us.
Yeah, now you have to go back. It's like you're looking for Easter eggs. Like when you watch a mystery movie and you're like, oh, yeah, exactly. Like, oh, now I see. You have to re-listen to it all.
It's like The Usual Suspects. This podcast has been full of foreshadowing for this episode.
The entire podcast is that bulletin board.
This episode was requested. Specific discussions about bones, as well as the methodologies we're going to be talking about, were variously requested by our listeners, specifically by Jerry, Diana, Jackie, Picklebore, EB, Kyla, Piero, Lydia, Richard, Kylie, Jordan, Rafa, Mark, Tim, and Sarah Grace.
Nice. Great request, everyone. You all knew how interesting this was going to be.
This is going to be so much fun. Hey, before we get into any of that, let's do some quick announcements. First and foremost, as always, this podcast is supported entirely by Patreon. Listeners who subscribe to support us on Patreon contribute to everything that we get to do with this podcast, from hosting to cool projects. And in return, they get access to all sorts of goodies like live streams with us and bonus noise and director's notes. And at certain levels, you get your name shouted out on the podcast. This episode, we would like to welcome and shout out Matthew, Andrea, Richard, and the EO Archaeote.
Thank you so much for your support, and welcome to Patreon.
Dear listener, if you would like to support us on Patreon and encourage our science communication efforts, find the link down in the episode description. Right now feels like an extra poignant moment to be reflecting on the support of our patrons, because we are actually recording this on the 8-year anniversary of Common Descent.
Yeah, it's today.
It is today. By the time you are listening to this, it will have passed, and also, our 8-year anniversary live stream will have just happened. We did it on February 1st. We hung out with our fans and we made some announcements of cool things that are coming up. Our theme for Silver Screen Science, a couple of new projects. Go back and watch that video if you haven't seen the announcements. The video is archived on our YouTube channel. We'll post some of those announcements on our social media. The most immediately important ones are, number one, we are doing a listener survey where our listeners will be able to provide us with feedback, information about yourselves, help us get to know our audience. And this survey also marks the return of the most important question in the history of our podcast. You, dear listeners, for the first time in a long time, will get to vote on snakes versus crocs.
Make your voices heard.
Also immediately relevant, if you go to our YouTube channel, there is now a new show.
Yeah, there is.
It is called Briks with Bask, and Will's making Lego builds based on podcast topics.
I've been having so much fun. I hope you all enjoy. please give your feedback and let us know what you like and what you'd like to see.
Check it out. Hey, speaking of cool stuff, we also recently were guests on the Gaming with Science podcast. Brian and Jason over there invited us to come on and play and talk about a board game called Holotype. We had a ton of fun. That episode should be out by the time this is that you can hear this. So we'll put a link to that on our website and in the episode description as well. Go check it out. We had a ton of fun talking with those guys.
It was so much fun.
And finally, we have a physical mailing address that sometimes people send mail to. Will, I understand that we have received lots of presents.
We have gotten so many neat things. We got a gift from Elizabeth that is a Year of the Snake gift bag.
Yeah.
Because it has a card, a bookmark, a magnet, and a neckerchief from the New Jersey Pine Barrens that says, tread lightly or else, and has rattlesnakes and ticks and poison oak on it, and it's great.
Excellent. Oh, they're also good, Elizabeth. Thank you.
Yeah.
I will treasure this snake paraphernalia all year and beyond. Will, when is the Year of the Croc? When's the Croc? Have a year on the Zodiac?
They don't have that, but they have the dragon, and since dragon myths have been connected a lot of times with the stories of crocs, I feel like, spiritually speaking. Anyways, we also got stickers.
There's a lot of places that refer to the word dragon is synonymous with the word serpent. So really, there's I'm 2 years saying, of the snake.
They obviously don't think so because they differentiated them, and the one they have is real small. All right, the one they have is really tiny. It's easy to overlook. We also got a bunch of really fun stickers, paleo stickers and animal stickers from August. There's a cicada one, which I cannot quote on here, but it is oh so wonderful. A grow, scream, reproduce, die. And
that's it.
And it is so great. So thank you for those. And finally, we got a book, Brian Aldiss, Hot House, from Debbie.
Thank you. We will be looking into it. I believe that book is a speculative fiction of a plant-dominated world.
Yeah, I read it. I read the back briefly, and it sounds... The first line, in a strange future, spiders over a mile in length travel the moon, is a
All right,
very...
We're on board.
I'm interested. And then we also got... This wasn't mail because it was hand-delivered. We got to hang out with Danielle the Bug Lover at the museum and walk around and see behind the scenes, but got gifts. Danielle made a black box with some preserved insects, beetles, and butterflies in it, which are so cool. And we got figures of Mosasaur and Caprasuchus, the boar croc, which was just so great. They both open their mouths, which makes all figures better.
Fantastic. Speaking of seeing people in person and getting gifts, I recently was given the gift of being shown around the Vancouver Aquarium by 2 of our listeners, Kayleen and Garrick, and it was delightful. So a huge thanks and shout out to both of them.
This is so cool. Thank you, everyone. We are being so spoiled, and you're all so kind, so thank you.
And now let us move on to the news from outside the podcast. Every episode, we like to talk about some new research and publications and studies that are being done out in our favorite realms of science. Will, what news have you brought to share with the class today?
Here in the Year of the Snake, I want to talk about a bone bitten by a croc.
This is unconscionable.
Hey, when we find one bitten by a snake, you...
Now, I don't appreciate that.
This is a juvenile pterosaur bone that seems like it was bitten by a crocodile.
That is very cool.
It's neat. This is research by Caleb Brown et al. In the Journal of Paleontology, and the article is by Will Dunham in Reuters. So, the bone in question is from an Azhdarchid pterosaur, so one of the big ones, that include Quetzalcoatlus and stuff, and is identified as a juvenile individual of the species Cryodracon boreas, which, just for a fun fact, that name means cold dragondom, the north wind.
Yeah, pterosaurs are pretty cool.
They get such cool names.
They get good names.
This is also, I didn't know this, one of the largest, like rivals Quetzalcoatlus.
Oh, interesting.
Adults reach sizes with wingspans of like 33 feet, 10 meters.
Oof.
So like big one. This one was not that size. This was a juvenile one. They estimate that its wingspan probably would have only been about 7 feet long based on its size.
Just a tiny pterosaur.
Just a little 2-meter wingspan pterosaur. Me and it could have hugged very easily.
Yes.
We would have had similar wingspans. This is from the Campanian Dinosaur Park Formation in Alberta, Canada, which dates to about 76 million years old. That at the time it would have been a warm, wet flood, like flat ecosystem. Where they said bisected with large rivers, so kind of, wetlandy-ish. The bone is a neck bone, so one of those long neck bones that they are so famous for in this group. They're often compared to giraffes when they would have been walking around. This is 2/3 complete, so it's not the entire bone, it's part of it, and measures at about 2¼ inch or 58 millimeters long. It's thin, and I liked this metric. The outer wall of the bone is less than a credit card in thickness.
Yeah, pterosaur bones, extremely thin-walled.
Just a very satisfying way to visualize that. They examined it under microscope and micro CT scans and found a conical puncture wound, 1/6 of an inch wide or 4 millimeters, so very tiny. Looking at the dentition and other traces of the potential trace makers, the potential biters of Dinosaur Park Formation. So the predators present in that fossil record, they found that it does not match the teeth of the predatory dinosaurs found here, and that the most likely candidate is crocodilian. There are a number of crocodilians known from the site, from this formation. Letosuchus, which is a around 12 foot, so 3.5 meter long crocodilian, and a smaller one known as Alberto champsa. There's also another predator that's croc-esque, but is not actually crocodilian. It's another group of reptiles called Champsosaurus that looks very crocky, but is outside the group. As is always the case with predatory traces, whether this was a killing bite or a scavenging bite, we can't know. Both fit well within the feeding habits of crocodilians.
Yes.
Like, they're predators, they're also scavengers. There's no signs of healing, which could mean that that's a killing bite or it was already dead. So what is kind of interesting is that feeding interactions involving pterosaurs are fairly rare globally. So this is a very nice finding just for the overall data set of things feeding on pterosaurs.
Yeah. Feeding traces in general are not super common in fossils. And pterosaurs have very delicate, thin-walled bones that are much more likely to be destroyed by getting bitten than they are for the bone itself to survive and then become fossilized.
They did make the point that I thought was interesting that in like this age and this kind of fossil site, crocodilian bite marks are not, as they put it, not uncommon. So like,
okay,
the fact that this is one of the very rare examples does mean that maybe pterosaurs weren't a regular thing in their diet. Like if it was, maybe we would see more, but definitely could have been, was a part of at least a crocodilian's diet. Another point that the article made that was just very interesting was that we don't know a lot about what Cryodracon would have eaten. We don't have any stomach contents from it, and it's been debated how these big Azhdarchids would feed in general. They made the kind of joking point that we almost know more about what ate them than what they ate, because there are other feeding traces on Cryodracon fossils from other predators at other sites. And within the dinosaur park. So like, we actually have a bit of info on what was eating them and their cousins.
So this is a species of pterosaur that is most famous for getting eaten by things.
Yeah, there is a velociraptor that had stomach contents in Mongolia with a relative of this group. Another specimen at this site that had bite marks on it. Another one from Romania that might also be croc bite marks. These were getting bitten a bunch.
These were the tastiest pterosaurs. Very cool. I think that it's very tempting in a case like this to think about, this is a large flying animal. So it makes the most sense to imagine that it was scavenged by a croc. But pterosaurs got a drink, and I think that
Yep.
it makes a lot of sense to imagine a croc could ambush and attack a pterosaur the same way that it could ambush and attack a zebra or a giraffe that comes to take a drink. But also, it is absolutely not unheard of for crocs to leap out of the water
Yeah.
and grab some. Now, I don't know how, I can't imagine a croc is going to like catch the trajectory of a pterosaur as it's flying overhead and jump up and grab it. But as a pterosaur is maybe taking off or is on the surface of the water or something, crocs are absolutely known to catch birds. And I assume
Yes.
bats, although I
Oh,
don't
yeah.
know an example off the top of my head.
There's actually a great video of flying foxes because they will wet their fur to drink once they get back in the tree, and freshwater crocodiles in Australia will gather and try to grab them as they swoop down to get water.
Whew.
And so, like, if they mentioned the fact that one of the feeding strategies that has been suggested for these pterosaurs is skimming, going along the water
Oh,
surface to try to grab
sure.
fish. So if they are feeding at the water, then that's Croc's bread and butter is taking advantage of the other animals that have to use the water.
Yeah. Now, I guess on the other hand, crocs tend to swallow their food whole. So there might also be the argument that if a single bone has survived with a bite mark in it, it might mean that this body was already coming apart and maybe it was scavenged. Or maybe two crocs got a hold of it and pulled it apart, and that one part of it floated away or something like that.
Yeah, exactly. This one tore a piece of the neck off and took it away and left a bite behind on
Yeah.
what eventually got buried.
Very cool. Well, as it turns out, my first news is also about diets and what ancient things we're eating, but in this case, the diets of our hominin ancestors, Australopithecus.
Cool.
This is research in the journal Science by Tina Ledecky et al., and we will link in the blog post to an article on Science News by Jake Bueller. Diet is an important feature of our ancestral lineage. Our ancient, our deeper ancestors were likely very herbivorous, eating a lot of plants. That's the case with most living apes. But at some point, our ancestors made the switch to eating much more meat. We see evidence of this in, tools and butchery marks, but also this is thought to be related to certain changes over the course of human evolution. We've talked about some of this in previous episodes, but our shorter faces and smaller teeth and weaker jaws compared to other apes and compared to our ancestors are thought to be linked to eating meat, which is an easier food to eat in many cases than plants, but also the suggestion that our bigger brains were able to get bigger because of the protein we were getting from a more meat-heavy diet.
Yeah.
So meat eating is thought to be an important part of Homo sapiens eventually becoming what we are today. But exactly when this dietary shift happened in our ancestry isn't clear. This study investigates the diet of Australopithecus. Australopithecus was a few species of hominin apes, very famous. Lucy is an example of a member of Australopithecus. These are the group that is the direct ancestors of our own Homo genus. Australopithecus has a lot of features that are very similar to modern humans, like the shorter snouts and bigger brains and walking on 2 feet. This study looked at isotopes. As we've discussed before, when we eat things, we are taking the chemistry of our food and using it to build our bodies. Different types of food have different ratios of isotopes of certain elements. So carbon and nitrogen come in multiple naturally occurring forms, and the ratio between those forms is going to be different depending on what you're eating, and so that ratio is going to get captured in our bones and teeth. In this study, they looked at carbon and nitrogen isotope ratios in enamel, so that's from teeth, from a variety of fossil mammals preserved in the Sterkfontein caves in South Africa, which are around 3.5 million years old. Among the mammals that they tested were 7 specimens of Australopithecus. So then they looked at those ratios in the Australopithecus teeth and compared them to the mammals living in the area and modern mammals.
Yeah.
And what they found was that the isotopes in the teeth of the Australopithecus specimens were mostly similar to herbivores, both modern species and the other species at their site,
Okay.
whereas they were quite different from carnivores. The isotopes indicate that they had a varied diet, so they were eating a variety of different things, but mostly it was C3 plants. So leaves, and as opposed to grass, leafy vegetation.
Yes.
This is really interesting because number one, tells us what Australopithecus was eating, that they were mostly vegetarians, but also because they already have a number of the features that we associate with modern humans that have been suggested to maybe be related to meat eating. They already have bigger brains and that shorter face, which suggests that the dietary switch might still have impacted the course of our evolution, but it does not appear to have been a one-to-one. Our ancestors started eating meat and then got big brains and weak jaws and stuff. Those
Yes.
processes were already underway.
Very interesting. It makes you wonder then what, were there other drivers for those features that are less obvious than that connection that would have been the initial benefit to start that process.
Yeah. The authors do note in the article that this they could still have been eating meat occasionally, right? This just shows that they weren't predominantly meat eaters. And they also made the very interesting point that this study doesn't rule out that they were eating insects or things like termites, which we do see. Like chimps are famous for this. They'll stick sticks down into the ground and pull termites and ants out. The authors point out that those are a protein rich diet, but wouldn't be reflected the same way in the isotopes. So that there is the possibility that they were getting lots of protein, but not from traditional carnivory. So they'd say the next step is to look at other hominin species, compare at other sites maybe, and try to get an idea of how did diets vary among hominins in space and time.
That's a really good point. And now it makes me wonder about other things, like do eggs factor into the protein the same way meat does? Or like, are there other high protein things that fall outside of herbivory but wouldn't show up with this type of study, or this specific study at least?
Yeah.
That's a very, very good point, and important to remember that it's not just there's not just two sides of the coin.
Yes.
Very cool. That's neat. My next news also has to do with human evolution, particularly our ears. We've talked about before that our ear bones are repurposed jaw bones.
Yes, episode 178.
This research may have found evidence that the cartilage of our ear was also repurposed from other structures and has potentially a very deep ancestral origin to it. This research is by Mati Tirupati et al. In Nature, and the article is by Sasha Pear in Live Science. So the ear and the inner ear have always been a focus of study just because they are such specialized features in mammals. Our inner ear bones.
And kind of important.
Yes, exactly. Especially to this here franchise that we are trying to embark on.
Yes, yeah, if it weren't for ears, common descent wouldn't exist.
Yeah, no, we are kind of reliant on them. Inner ear bones are a very much mammal thing. Other animals have similar structures, but our three bone system is a unique structure to us. Other aspects of our ears have also been wondered, how unique are these two mammals, or how do they have similar origins to those inner ear bones where they came from a past structure? The outer ear, our big ear flaps, are also a mammal evolutionary trait. Famously, that outer ear is structured by cartilage. It is supported by cartilage inside. That's what lets it bend but still retain its shape. The evolution of this is difficult to study because cartilage does not typically fossilize, so we do not get a good idea of how it developed and where that cartilage might have come from. So we have to look at it from a genetic point of view. Particularly, the cartilage in our ear is a subtype called elastic cartilage. It's more flexible than like haline cartilage or fibrocartilage, which is the kind found in your nose and between your vertebrae, respectively. And in general, there's very little info about elastic cartilage outside of mammals, period. So we don't know a lot about how it's related to other animals in general. They wanted to look at, does it also have an ancestral origin, particularly fish, because that's where those jaw bones originally came from, were our fish ancestors. Does this have any connection to fish structures? So they used protein stains. That's where you stain tissue and the stain hangs onto particular proteins or molecules, so that way it only colors the type you're looking for and shows you where it is and if it's there. They use those on the gills of zebrafish, Atlantic salmon, and 3 other species of fish, and found that they do contain elastic cartilage. So we didn't know that. Now we do, that it's found in fish, at least this variety. These are all bony fish, which are your typical fish that you think of, not the cartilaginous like sharks. And due to the spread, suggests that it's likely a feature of the group, that it is something found within them, at least commonly. They wanted to look at the evolutionary link between our elastic cartilage and their elastic cartilage, so the gills and the outer ear. They looked for gene control elements called enhancers, which are DNA sequences that can activate other genes when bound with specific proteins. These are tissue specific, so the genes are very tightly tied to the tissue or body part that they activate or affect. So they tested whether or not these enhancers from a fish are similar to the enhancers in our outer ear by inserting our enhancers into a fish and seeing if it activated their gill arches in a way, and they did. The experiment was able to trigger activity in the gills of zebrafish with our outer ear enhancers.
That's it's very cool, because it's like, yeah, we took the turn the ears on switch and we put the ears switch in a fish, and when we flipped it, their gills turned on.
Yes.
Cool.
That evidently that plug is the same genetically.
Yes. Now, obviously, that's simplified, but it's this specific type of cartilage seems to have the same genetic system.
Exactly. And when they reversed it and went from zebrafish gills to mouse genome, it activated within the mouse outer ear.
Interesting.
So the cartilage within our outer ear and their gill structure seem to have very similar genetic pathways and genetic mechanisms. They did another experiment with tadpoles and the green anoles, so amphibian and lizard, and showed that they also inherited the tadpole's gill structure, and the ear structures in the lizard also had a similar connection to fish.
Oh, interesting.
So it seems very likely that the elastic cartilage that fish use to support their gills has been repurposed in multiple groups for similar and different structures, leading to our outer ear using it in its support. So the little bones came from a fish jaw. It looks like the cartilage of our outer ear came from their gills. This means that if this cartilage is all connected, with the lizards in the mix, suggests that elastic cartilage began migrating from gills to outer ear at least by the time reptiles appeared in the evolutionary record, so around 315 million years ago at minimum, which suggests a very ancestral link to all this. And it may go even further back, because they also did a test with horseshoe crab gill cells. They have book lungs in their gills, and it also triggered activity in zebrafish gills. So it may be that this elastic cartilage, this type of structure, goes back to very deep origins in animals.
Interesting.
And that we're building off a similar pathway to make these different structures that are kind of similar, but not all doing the same job anymore. There's still a lot to confirm with this. There's definitely could be cases of convergent evolution, but the fact that the genes are so similar definitely gives a lot of indication that there might be an evolutionary ancestral connection to the gene system we're seeing here. But it does potentially mean that the thing that let us build our outer ear came from ancient invertebrates
Yeah.
building their gills.
It's fascinating because obviously every part of our body is developed from some ancestral part of something. I'm
Yes.
always really fascinated when we find out the genetic program that makes this goes back. So this was a genetic program that built, some of the gills in like
Mm-hmm.
early invertebrates, and all of us have just been using a repurposed version of that program. That
Yes.
this isn't a program that mammals developed on our own, or even that fish developed on their own. This might just be one of the core genetic tools of animals, period.
Yes.
And that's one of the ones that we use it in our ears.
Yeah.
Super cool.
These always feel funny to me because it's like genetics we typically emphasize of like it's not that it's always more complicated than we're simplifying it down to. But here it really is that, yeah, if you take this enhancer and plug it into a bunch of different animals, it still works. It's doing different jobs, but it's still functioning.
Well, my last bit of news has very little to do with what you were just talking about, but a lot to do with my other news. So, this is also using isotope analysis to determine the dietary signature in different animals. But in this case, it's looking at what do dietary isotopes look like across reptiles.
Ooh.
This is a cool one. You're going to like this one. This is research by Michael Weber et al. In Proceedings of the Royal Society B, and we will link to an article in earth.com by Andre Ionescu. As I mentioned before with the Australopithecus study, isotope analysis is commonly done in both fossil and modern species to find the dietary signal that animals are picking up from their food. In that previous study that I mentioned, they were looking at carbon and nitrogen isotopes, which are extremely commonly used for that. This study is looking at two different isotopes, calcium and strontium, also useful. Also, the ratio differs depending on what the animal is eating, but typically, the authors note, studies on these isotopes are done with mammals or aquatic habitats. So
Yeah.
there isn't a lot to go on with modern land dwelling reptiles, which means we just don't have a good foundation of what do these isotopes do in the bodies of reptiles. What does the signal look like? Which is important to establish if we want to be interpreting the diets of ancient reptiles.
Yeah.
So here they did calcium and strontium analysis on both tooth and bone in living reptiles with known diets. So we can say, all right, we know what this species eats. What do their isotopes look like? They looked at 28 species of lizards and crocodilians with a variety of diets, plants, insects, meat, and so on. Notably, they left out turtles and birds because they don't have teeth. There was no mention specifically of snakes that I noticed in the article, which I assume is just an egregious oversight on the part of the editors. This allows them to establish a reference of how these isotopes manifest. They found a whole bunch of interesting stuff, but I'll mention a couple of the highlights. For one, both calcium and strontium do track different diets, just like we see in mammals. They also track different positions on the food web, so different trophic levels where the ratios are at one extreme in insectivores and then into herbivores, and then the other extreme is carnivores, the level of that ratio. So you can actually follow how high in the food web is this animal.
Cool.
That is also something that's similar in mammals, although they noted that the specific values they're measuring are different from mammals. So there is some physiological shift. One thing that was really interesting is that two of their reptile groups were their carnivore representatives. They had monitor lizards and they had crocs.
Yeah.
And they noticed that the isotopes were carnivorous isotope ratios for both of those, but they were different between the monitors and the crocs. They noted specifically that if we're thinking of the ratios as like higher in the trophic levels, the crocs were as far above the monitors as the monitors were above herbivores. And
Yeah.
they suggest that that probably is a reflection of the fact that monitors eat a variety of, they'll eat big things and little things and bugs and stuff, whereas crocs tend to be apex predators
Yep.
eating large whole meals.
Yeah.
So their analysis was even able to differentiate between different levels of predation between these reptiles.
That's really cool.
They found that, generally speaking, bone and teeth were very similar in their isotopes, which apparently is different from mammals, which is thought to be related to the fact that mammals get milk, which might be messing with those isotope ratios.
Oh, that makes a lot of sense.
But they did notice that an exception was Heloderma, which are Gila monsters, beaded lizards. Their Heloderma specimens had a very different ratio with calcium between bones and teeth. The teeth reflected a diet of eggs, which is correct. That's what those lizards were eating. And so it's like, all right, cool, eggs, that makes sense for what you'd see. But the bone isotope ratio was more of a carnivorous signal. And the authors point out that in this case, that might have to do with how the animal's diet changed as they grew up. Because bones last essentially the whole life of a reptile, especially if they're short lived, but teeth are replaced on a weeks to months long cycle. So the bones might be capturing your lifetime diet, and your teeth might be capturing more of a recent diet.
Oh, that's very cool.
So it could be that these lizards, when they were younger, were eating more small animals and being carnivorous, and then switched to a specialized eggs diet as they became adults. And that might be where that difference is coming from.
Yeah. The idea of getting two different life signals is very cool. Something about that is deeply satisfying to me.
Yes. As part of this study, they also looked at microwear, which is microscopic signs of damage on the teeth, and they found that combining microwear and isotopes, they were able to differentiate things like egg eaters from carnivores or egg eaters from durophagous lizards and reptiles that were like eating hard-shelled foods and stuff. So this study suggests that there is a methodology to look at damage on the teeth and isotopes and put them together to get a very accurate indication of what these animals were eating, which could potentially help identify changes even throughout their lives or over evolutionary lineages in the fossil record, which is especially important for interpreting the diets of ancient reptiles, which we like to do quite a bit.
Very cool. Man, like, I didn't know that we didn't know with reptiles. So
yeah,
that's cool. And so cool that we are able to get really refined detail, like surprisingly detailed insights into their lifestyle and habits and even ecological roles.
Yeah.
That's an awesome study.
The paper also makes a note. This, I think, is like way late in the paper that says a lot of the time when we are interpreting the diets of dinosaurs using similar studies, we're using comparisons with modern mammals because that's where the data is. This study found, comparing just some information that was already out there from other studies, it looks like dinosaur isotope signatures are more like modern reptiles than they are like mammals. So with this kind of data, in theory, this will help us be yet more accurate with our interpretations of dinosaur diets.
Nice. That's so exciting. It's also exciting because it feels like this is the beginning of many more studies. And that's always a fun moment to get to see of like, this is kind of the first time this is being done on a wide scale.
Mm-hmm.
So hopefully we will see either these researchers or other researchers go, okay, well, now we're going to do snakes, or we're going to do this group of lizards. We're going to start adding on to that data pile, and now we'll have a new database to use.
Yes. So very cool. It's very exciting to see studies. This is a modern study, but with broad applications to the fossil record.
Very cool.
And with that, I am delighted to see that so much of our news was related to the skeletal system and bones and stuff of fossil animals, which, of course, it's hard for it not to be unless we fill the news with invertebrate discussions. But that means that now we are done with the news and we can move to our main discussion, where we will be talking about bones. And we're going to start broad and then get very delightfully specific on how bones work and where bones come from. So if you've been waiting for 8 years for us to finally explain what we're talking about, just after a short musical interlude, stay tuned. In paleontology, when we talk about the tissues in an animal's body, we very commonly refer to soft parts and hard parts.
Yes.
The soft parts being the, skin and muscle and protein and hair and stuff like that, the kind of stuff that doesn't fossilize well.
Mm-hmm.
And the hard parts being in invertebrates, shells and exoskeletons and spines and stuff. And in vertebrates, hard parts almost exclusively means teeth and bones.
Yep.
In paleontology, we really enjoy those hard parts of the body because we get them as fossils and they can provide us all sorts of cool information about the ancient animals. The key difference between the soft parts and the hard parts is that the hard tissues are mineralized.
Yeah.
That means that most of the tissues of our body are made up of proteins and organic molecules of various sorts. Hard tissues incorporate minerals into that tissue structure. In our bodies, the three sort of big deal famous hard tissues are enamel and dentine,
mm-hmm.
which are tissues in our teeth, and bone. Bone, specifically, the mineral in bone is calcium phosphate. We'll talk a bit more about that in a little bit. So bone is the major part of our body that is built of mineral components.
Mm-hmm. Mm-hmm.
This is another one of those topics that we've mentioned on a handful of previous episodes, that the idea of introducing to people what a bone is feels sort of superfluous. They are core to the way that our bodies work. They are literally the scaffold upon which the vertebrate body operates.
There's a reason that when we have like undead, skeletons are a perfectly suitable stand-in for an undead person.
Bone in vertebrates typically performs three major primary functions. The first is physical structure and support. Bones are what give our body its shape. They hold up the whole body. They also form the anchor points for our muscles, right? The way that muscles work is they're contracting, they're pulling. Well, they have to be pulling against something. And what they're pulling against is the hard substrate of our skeleton.
Same way that buildings have scaffolding and cars have a steel frame, you need a foundation to build off of, and that's what our skeleton is.
Yes. It is not a coincidence that the only animals on land that get bigger than the size of a large crab, I guess, are vertebrates who have bones to support. It's like wood for trees.
Yes.
The second major function that bone provides is protection. Bone is very rigid, it's hard, it's solid. Bone protects our internal organs, right? Our brain, arguably our most important internal organ, is completely surrounded by solid walls of bone. Our heart, our lungs are within our rib cage. Inside of bones, there's cavities for bone marrow, sinus cavities, vasculature, all sorts of stuff like that. In some animals, they've taken this a step further and actually developed bony armor.
Mm-hmm.
Right? Crocs have bony armor. Turtles, are probably the most iconic example of this, who have moved a whole bunch of their skeleton outside the rest of them.
Yeah.
And third, and this I think is the sort of unsung last primary function, is that bones serve as a reservoir of minerals.
Mm-hmm. Mm-hmm.
Minerals and certain elements, certain ions are really important for the way our body functions. Like calcium and phosphorus, which are major components of bone, are both important for the way that our nerves function and the way that our blood functions. By having an internal scaffold that is made of those minerals, we're carrying around an extra storage of those components.
Yeah, same way that we have fat storage for, when we need extra energy and things like that. Bones are also a storage. They just also have a bunch of other jobs.
Yes. Well, and we don't want to pull too much from them because of the importance of those other jobs. There are a bunch of examples of times where animals will extra drain their bones of those mineral resources. We've talked about on the podcast before, birds will pull calcium out of their bones when they're preparing to lay eggs because they're repurposing that calcium for eggshells. There are also examples in fish where they will start pulling mineral from their bones during migrations.
Oh, that makes sense.
That extra bit of fuel to keep the body running properly. And then outside of those major functions, bones have been adapted and evolved all across the vertebrate family tree to do all sorts of wacky other stuff. There are animals that use bones for display in, head crests and stuff like that. There are animals that use their bones as weapons.
Mm-hmm.
Like in the case of horned animals that we have today, or tail clubs that are made out of bone in things like ankylosaurs. There are animals whose bones are used for communication, like the head crests of hadrosaur dinosaurs, or, as we discussed in episode 178, our inner ear bones.
Yes.
Whose function is to transmit vibrations that we pick up in our ears and then turn them into something our brains can interpret.
Yeah. And bones are at the core of so many physical adaptations. Like, if you change something about your arms and legs, you have to change your arm and leg bones, and et cetera, et cetera.
So bone is this very fundamental aspect of vertebrate life. It is the defining thing about vertebrate life. It's the things that have vertebrae. Now, we'll get into some of the evolution and utility of bones, but first, some anatomy. Let's explore some details about what bone is. So bone is a type of tissue, right? Our bodies are made up of tissues. There's different types of tissues throughout our body. Bone, specifically, is a type of connective tissue. Connective tissues are tissues that tend to exist in between other tissues.
Mm-hmm.
They often are offering support or protection to those other tissues. Connective tissues tend to be made of cells surrounded by an extracellular matrix. That basically, it's cells that are surrounded by stuff.
Mm-hmm.
The stuff is also often fibrous organic material, a gel-like ground substance. Connective tissues include things like tendons and ligaments. Blood is also technically a connective tissue whose extracellular matrix is fluid.
Yeah, that's a good point. Huh.
Mm-hmm. In bone, specifically, the matrix is, very simplistically speaking, two parts. An organic part, which is made up of proteins and stuff, especially fibrous things like collagen, and the inorganic part, which is calcium phosphate, which is mineral. It is a form of apatite, specifically hydroxyapatite. Sometimes it's referred to as bioapatite.
Ooh, cool.
It is the form of mineral appetite that is created by and within the bodies of vertebrate organisms.
That's cool.
This combination of fibrous protein and mineral component makes bone hard, rigid, but not brittle.
Mm-hmm, mm-hmm.
Within that matrix of mineral and organic components, there are also lots of cells, there's blood vessels and nerves. Bone isn't just solid mineral. There's all sorts of stuff going on in there. It is a tissue. It is an actual tissue with lots of organic components. This is as compared to things like dentine and enamel in our teeth, which are also heavily mineralized, which are also tissues, though they vary in what kinds of organic material is in there, how mineralized they are. Enamel, which is the thing that coats our teeth, is super mineralized.
Yeah.
It is extra sturdy. It is the hardest material that our bodies create.
Yeah, it's intense compared to everything else.
Inside of a bone, so zooming out a little bit, if you take a bone and you break it in half, so if you've ever done this with like chickens are not the best example because bird bones are weird, but if you break a bone in half, there are, broadly speaking, two types of bone tissue, two arrangements of bone tissue. There's cortical bone and there's spongy bone.
Mm-hmm, mm-hmm.
Cortical bone, also called compact bone, is smooth and dense, and it is what forms the outer wall of bone. So if you break a femur in half, there's this thick outer wall, and then the inside is this spongy looking texture. That outer wall is compact bone. Compact bone is highly resistant to stress and strain. It is extremely important for that support function. It's also very dense.
Mm-hmm.
I've seen it noted in a couple places. Cortical bone makes up about 80% of the mass of our skeleton.
Yeah.
So if you took all the spongy bone out, you would still weigh almost as much as you do.
Which is pretty intense.
The interior structure, spongy bone, also called cancellous or trabecular bone, this tends to form the interior of a specific bone. It is, as the name implies, spongy. It's very porous. It's much less dense. It's also less rigid. There's more elasticity to it. And there's lots of space in there. Spongy bone, interestingly enough, mineralogically is the same as cortical bone. There isn't like the mineral structure of it is not different. It is just the physical arrangement of the tissue.
Well, it's like how a steel beam and a steel wire could be made out of the same steel, but you're going to have very different physical properties if you build stuff out of them.
The cavities and spaces, those pores within the cancellous bone, the spongy bone, make room for other tissues. Most famously, marrow.
Yes.
Red bone marrow is the tissue that produces red blood cells, white blood cells, and platelets, which are all extremely important for our immune system and our circulatory system. There's also yellow marrow, which is fat storage.
Mm-hmm, mm-hmm.
So bone is also a production factory for like bone contains the tissue that produces blood cells and stuff.
Which is always a weird thing the first time you learn it, because the sentence "your blood comes from your bones" sounds like the start of a spell. Like the blood of my bones. Well, that's all your blood. Like
yes, that's where they come from.
That's how we get it, which is pretty awesome.
It makes a whole lot of sense. We mentioned that protective benefit of bone. If you're going to put your red blood cell and white blood cell factories anywhere, putting them inside the mineralized tissues, it seems like a great idea.
Yeah, that is a fundamental, you need blood to die. Bleeding out is very bad.
That's bad. Keep that inside your body.
Yes, so absolutely, it makes sense that this is a critical thing. Like we put our brain inside bone, putting that inside bone makes sense too.
Yes. There are other types of tissue within the structure of bone. Periosteum, which comes up a lot in discussions of bone anatomy, is a fibrous layer of cells that covers the outer surface of bone. So it's sort of
Mm-hmm.
this extra tough layer. Endosteum is a similar layer on the interior of bone. There's a lot of complexity to the tissue that makes up bone. There are also different types of bone. Now, as usual, trying to classify, put things in boxes in nature does not always work. I have regularly seen it described that there are, generally speaking, three types of bones: long bones, short bones, and flat bones. Long bones are the ones that tend to be long and round. Our leg and arm bones, our ribs. Short bones tend to be things like our wrist and ankle bones, our carpals and our tarsals. And flat bones are things like our skull, our shoulder blades, our pelvis. These differences reflect differences in the where those tissues are, right? When you
Yeah.
break an arm bone in half, there is an outer circular wall of cortical bone, whereas in a flat bone, you've got these parallel walls of cortical bone and then a thin layer of that spongy bone in between.
Mm-hmm. Mm-hmm.
Those are all the bones that typically get counted when we say, here's how many bones there are in the human body. But in reality, there's way more bones than that, because there are also intrasutural bones, which are little bones that exist in between the sutures of our skull, for example, where multiple quote, real bones with names, connect to each other. There's these little bones in between that help form the suture. There is also a very famous category of bone called sesamoids,
Mm-hmm. Mm-hmm.
which are a type of bone that forms at joints. Typically, they'll form within tendons. They form at joints. These can be extremely variable. They're all over your body. They come in all different shapes and sizes. They are inconsistent in where they show up and when they're present, not only in individuals, but across species. So, the 206 bones that are in our body is an underestimate. You've got more bones than that.
Yeah, we just don't. It's like the small asteroid bodies in our solar system, where it's like
Yes.
most of those don't have names because they're too tiny and they're too hard to track.
Yeah, and who knows how long they'll exist.
Yeah, so there's roughly this many of them, and they are usually around this shape and size. And yeah, there you go. But then here's the things we've named.
Yes, exactly. That's kind of how our bones work. Another important aspect of bone anatomy are bone cells.
Mm-hmm, mm-hmm.
These are specific cells associated with bones. There is a lot of detail here, but I'm going to simplify it to the general categories. Osteoblasts are a type of cell that makes bone. They produce the organic material, the proteins and stuff, and then they release the minerals and they facilitate the mineralization process.
Yeah.
So they put down the soft scaffold and then they mineralize over it to make bone tissue.
Which is very satisfying because, like, that's how we make buildings. Is we come in and put down walls and scaffolding inside of it and rebar, and then we fill it with concrete. And like it's the same steps just inside your body.
Yes. Now, if the analogy were truly appropriate, what would happen next is that the construction workers who do that would be surrounded by their own concrete.
Yes.
And stuck in there forever. Osteoblasts become osteocytes, which is a bone cell that basically isn't creating bone anymore. It's just trapped in there, trapped within the matrix, sitting within a little cavity inside the bone.
Because your body is a Magnus episode where horrors are bestowed upon your cells all the time.
Interestingly, these osteocytes sitting in their little cavities are in communication with other cells through little channels of cytoplasm that go throughout the bone.
Interesting.
And then there are also osteoclasts, which are cells that dissolve bone, that break down bone. Those are important because bone is constantly being renewed and replenished. This is maybe the most unintuitive part about learning about how bones work. Bones are living tissue.
Yes.
It's not just rocks that are sitting there doing nothing. Bones are active, they are full of cells, and the bones that you have now are different than the bones you had a year ago.
Yeah. Like you are replacing your bones throughout your life. So it is not the same material that made up your skeleton when you were a child.
Yes, just like your skin, just like your other
Mm-hmm.
tissues. But bone operates in a different way. This is a process called bone remodeling. The reason it operates in a different way, right? Most of the tissues in your body replenish and rejuvenate through the simple process of cell multiplication, right?
Mm-hmm.
Cells dividing and making more cells, and apoptosis, which is the process by which cells are destroyed.
Yes.
So you clear the old cells, you multiply to make new cells. Bone is mineralized. So it isn't just a sea of cells the same way that a lot of our other tissues are.
Yeah, mineral doesn't die.
Mineral does not die. Minerals is forever. So the way bone remodeling happens is that those osteoclasts will dissolve a section of the bone, a microscopic section of bone, and then those osteoblasts make more bone. So a section of bone will get replaced at a time. The mineral gets dissolved and reabsorbed. It's called resorption. Those minerals are recycled back into the body, and then new mineral tissue is placed down. This is really important because it maintains the shape and quality of the bone. It repairs wear and micro fractures and stuff that build up over, because bones are, they're doing a lot of work.
Oh yeah, like when you exercise and stuff, famously that's how you build muscle, is the breaking down and then rebuilding of muscle. You're doing damage to your muscle when you work out, and healing that damage is what makes you stronger. Your bone gets microfractured especially if you're doing any like contact sport, like martial arts or football. Like you're getting small cracks in your bones constantly.
Yes, and even just walking around,
Yeah.
sitting, your bone is being worn. Remodeling can also change the shape of your bones if needed, right? But this is something that we'll see where, like, if a person or an animal is favoring one side of the body over the course of their life, the bone will change shape to support that stress. And remodeling is also recycling those minerals. That process of resorption is essential for getting the calcium and phosphorus and other trace elements out of the bone when it's needed. Indeed, when bone breakdown and buildup are not balanced, most often what happens is that new bone isn't being formed as quickly as bone is being dissolved and resorbed. That's how you get osteoporosis.
Yep.
Where the bone becomes weaker and more brittle because it's becoming less dense effectively.
Yeah. Well, and you also need cells that can break down bone in case you get like cancerous growth or something where bone is growing in a way it shouldn't. That's why we need cells that can kill cells, because sometimes there are cells in our body we need to die. Yes. Bone can be the same way.
Well, and also bone is able to heal.
Yes.
Right? A broken bone, outside of those, the wear and tear, if you break a bone in half, your body can heal it by putting down new bone. We're not going to get into that in this episode because that's a whole other topic. But this is a tissue your body is able to produce. All of this underscores the important thing to keep in mind about bone, which is that bone is active. And what that means is if you take a really close look at bone, there are signs of that activity, of how that bone has been growing and changing over time. And that is very important for biologists studying things and also for paleontologists. More on that in a bit. Bone in our body generally forms one of two ways. Again, speaking very broadly about, I found a lot of papers that were like, here's 100 pages about the details of how bone works.
Yep.
I am simplifying for your benefit, dear listeners.
And for me.
And for Will's and for my benefit, because I don't understand half of that stuff. Generally speaking, bone forms in two ways. Either mineralizing within a membrane, so like inside skin or inside another membrane, This is often called dermal bone within the skin or membrane bone. Many skull bones form this way. Osteoderms, famously, the armor and things like crocs and armadillos, form within membranes. The other way that bone forms is it mineralizes from cartilage.
Yes.
That cartilage is placed down first, and then that becomes replaced and mineralized to form true bone. That is the case with most of our bones. These are not two separate processes. There's overlap and there's in-betweens and there's all sorts of, but those are kind of the main two things, which useful to know for getting into the evolutionary history of bone, but also introduces us to another thing that we have to talk very briefly about, and that's cartilage.
Yeah.
When planning this episode, I wasn't sure how much this was going to end up being a cartilage episode. But I said, I'm pretty sure that if you're going to talk about bone, you have to talk about cartilage. Cartilage is another form of connective tissue. Unlike bone, cartilage tends to be thin. It's flexible. It's not mineralized. Cartilage tends to not be porous the way that bone is. It also typically is avascular. So there aren't blood vessels. I think for the most part, there's also no nerves within cartilage. It isn't quite the living tissue the same way that bone is. Cartilage matrix, so again, this is connective tissue. It's cells within a matrix. The matrix is made up of fibrous proteins, often collagen. Collagen is a very famous, we talk about collagen all the time. Other organics, but again, lacking usually that mineralized component. Sometimes you can get mineralized cartilage, but for the most part, it remains soft. Cartilage tends to function as support for other tissues. Cartilage, even though it's not bone, it's still stronger than most tissues.
Well, it's, once again, if you've ever eaten like a chicken leg and you get to the cartilage piece, you can chew it, which you can't do with bone, but it is not fun to chew. No. Like, it, or at least you're going to notice when you accidentally start chewing a piece of cartilage, because it's hard.
It is sturdy and it is resilient. It's not rigid like bone, but it is still a protective, supportive structure. Cartilage also functions to help connect other tissues to each other. With bones, cartilage often exists near and around bones as shock absorption and to reduce friction at places like joints.
Yeah.
So our joints have cartilage in them and they help our bones to move more smoothly past each other. Our bodies are full of cartilage. Cartilage exists outside of bone. They are not just like two different versions of the same process. They're doing different things. Our rib cage has a bunch of cartilage in it. Our windpipe is supported by cartilage. The discs between our vertebrae. So if you hear somebody say they slipped a disc, that's a cartilaginous disc that exists between the bones of our backbone. Cartilage is also, as I just said before, commonly a precursor to bone.
Yeah.
Developmentally, there are many times where cartilage will do the job of bone until it's time for the bone to get there. In us, when we are developing, when invertebrate animals, including us, our skeleton during early development, like fetal development, our skeleton is mostly cartilage.
Yeah.
And then it mineralizes into bone as we develop.
For a very short time, not so different, you and I, to sharks.
Yes, and sharks are an excellent example. Lots of animals have cartilage for all different purposes. Cartilaginous fish, like sharks and rays, typically have a full skeleton of cartilage, and in their case, it often is mineralized.
Yes.
So it isn't their bodies aren't just like flimsy cartilage. They're not all made of like our earlobe material. There is mineralization to their cartilage. So they don't have bones, but they do still have that sturdier structure to their skeleton.
Yeah.
Cartilage also is not unique to vertebrates. Cartilage is actually very common in other animals. In arthropods, many arthropods have cartilage in their gills and their breathing tubes to support them. Snails have cartilage that supports their tongue, the radula. Cephalopods have cranial cartilage
Yes.
that supports like the shape of the eyes or the shape of whatever the thing is, the mantle in
Mm-hmm.
squids and octopuses. It appears that cartilage is a convergent feature, that cartilage
Yeah.
has evolved over and over again across animals.
I knew about cephalopods because that's like famously you'll hear with octopus that they can fit through any hole their beak can fit through, but truly it's the cartilage ring that supports their brain that is the defining factor of how much they can squish. I didn't know about arthropods and snails.
And those are just like the examples that I pulled.
Yeah.
Cartilage is all over the place.
Cool.
Which gets us into this next question, which is, where did bone come from? The evolutionary history of bone. This is again, essential part of the vertebrate body plan. The origin of skeletons, the origin of bones is not only an important part in the evolution of vertebrates, right? Obviously, all vertebrate diversity is built upon this literal foundation of bones. It is also important and essential to our understanding of vertebrate evolution because before they had bones, we didn't find them in the fossil record very easily.
Yep.
Now, just like cartilage is a very common feature across animals, so is the mineralization of tissues. This process, biomineralization, animals building minerals with their body processes, is extremely common. Tons of invertebrates do that, right? Clamshells are that way, snail shells are that way, the skeletons of coral. These are all ways that animals are using minerals to build bodies for themselves. This is also a very ancient practice. The oldest mineralized tissues in the fossil record go all the way back to at least the Ediacaran period.
Yeah.
More than 550 million years ago, before the Cambrian explosion, we have early fossils of sponge-like animals or coral-like animals that have mineral components in their tissues. As time goes on, we start to see it in things like worms and arthropods that are building shells and spines and internal skeletons and exoskeletons.
Yeah, mouth parts and stuff.
Yeah, absolutely. For the most part, these other animals tend to be making calcium carbonate structures, the minerals calcite or aragonite. It has been, I found one study that was examining sort of the ancient history of mineralization and points out that there are so many different lineages that use biomineralization with varying structures and chemical compositions that it seems to be a convergent feature, that the ability to mineralize things has evolved multiple times, but there's enough similarities that it might be that the toolkit that they use to do it is ancestral.
Yeah.
That the capacity to manipulate minerals and elements in your environment in that way might be an ancestral feature that multiple lineages have then taken and turned into building their bodies out of minerals.
Which makes sense.
This might also be the case for bone. The earliest bone or bone-like structures go back to the Cambrian period on and around the base of the vertebrate family tree. So we go way back to the earliest vertebrates and we see things like there's a group called the Miloconmingidae, which includes famous things like Haikuichthys, which are these sort of flatfish kind of looking creatures. They don't quite look like fish that we would think of today, but they have fins, they have eyes, their muscle structure is similar to vertebrates. So these are considered to be either very early vertebrates or very closely related to the first true vertebrates.
Yes.
Haikuichthys and some of its relatives have some mineralized parts of the gills and parts of the fin, like the spines that support the fins. These appear to be cartilage or something similar to that, sort of an early limited version of mineralization. We also see early cyclostomes, which is the group that includes today's lampreys and hagfish. Both the modern ones and also ancient ones have cartilage that supports their oral disc, so their suckers on their face, which is sometimes mineralized. And there are also, in the fossil record, conodonts, which are a very famous early portion of the vertebrate family tree. Conodonts are these eel-like or lamprey-like creatures that are famous for their teeth, question mark. They have these tooth-like structures in the mouth and throat that are made of enamel-like tissue and dentine-like tissue.
Yes.
So we've got these like early vertebrates or almost vertebrates that are experimenting mostly in the mouth and the gill regions with early forms of mineralized tissue, some of which seem to be very much like some of our dental tissue that vertebrates have today.
Which is always so interesting when it's that early on in the lineage that we didn't end up at it. We kind of, that started out as a thing defining our group.
Yeah. After those, we move into sort of the true, these are for sure vertebrates, a whole diversity of groups that are commonly called jawless fish. A lot of these early famous, like Roomba or Hoover looking fish. Some of them have fins, some of them don't. There's a whole bunch of weird fish-like creatures.
They really do just look like undersea hand vacuums.
Yes.
Like that's very much the shape of a lot of them.
Many of them have scales or plates on the surface of the body, right? Sometimes over the head, sometimes over the other parts of the body, sometimes very extensive. And these variously include enamel-like tissues, dentine-like tissues, and bone.
Yeah.
Calcium phosphate bioapatite as we know it-esque, similar to bone today. Some of them also have fin supports or other internal tissues that include calcified cartilage or similar mineralized tissues. The earliest bones that we see when we start to see true bones are acellular, so they don't have those cells inside of them. That's something that then develops a little bit later. So in these early jawless vertebrates, it's very tempting to think that, bone evolved like, where there was a time where there was no bone and then there was 10% bone and then there was 50% bone. But instead, there's this diversity of playing around with different minerals and different parts of the body to mineralize.
Which makes complete sense, as we've already talked about the fact that like you can have degrees of mineralization and different amounts. So like it makes perfect sense that some organisms were like, all right, I'm only going to do 15% mineralization because I just need it to be a little tough.
Sure. Yeah, or I'm only going to do it in the mouth.
Yes. So
right. Yeah, I support my mouth and support my fins, but that's it. And others were saying, no, we're going to cover the whole head with this stuff. This stuff is great.
It is an adjustable ratio of how much you're doing with it. You don't just have to have bony skeleton or not.
From those early jawless fish come the jawed fish. Right? Now we're talking familiar looking vertebrates. Things like early bony fish and placoderms and cartilaginous fish. These feature more mineralized tissue. They have proper jaws and teeth. Some really famous early examples are placoderms, the armored fish, which we talked about in episode 29, who are covered in these bony plates. Often these are bone, like proper bone, with enamel and dentine-like tissues associated with them.
Yeah.
By this time, in placoderms and other early fish, we're also starting to see the beginning of cortical and spongy bone. The differentiation between the different types of bone. We get early cartilaginous fish. Now, I mentioned that our modern cartilaginous fish have skeletons of calcified cartilage. This appears to be a reversion in cartilaginous fish, that the earliest cartilaginous fish weren't as cartilaginous and had more of a bony internal skeleton, and they have lost their boniness over time.
Which is so the opposite of what, if you were just asked to lay out the order of steps that bones evolve, it would be very easy to go, well, I know sharks were some of the first jawed fish. So, and they have a cartilage skeleton. We already talked about the fact that cartilage can turn into bone. So that makes
Nice
perfect sense. Nope.
They had bone and they said, nah, who needs it?
Which is great.
And you get osteichthyes, the bony fish, which are most of the fish that we're familiar with today, which have a more extensive internal skeleton, including, the pelvic and pectoral girdles, more bones of the skull. So there is this time throughout the early Paleozoic where there's this diversity of bone and bone-like minerals forming scales and spines and rays and teeth and jaws, different types of bone. So that dermal, membrane bone and that sort of cartilage related bone. Often these bones are alongside dental tissues or alongside, calcified cartilage. There was this, this isn't one of those cases where there wasn't bone and there was. There was a period of tens of millions of years where vertebrates tried out a bunch of stuff and settled on the kinds of bone diversity that we see in modern fish. And then eventually, when they give rise to land-dwelling vertebrates, they narrow it down even more.
Yeah.
Right. In us, our dental tissues are only in the place where they're named for.
Yes.
We keep those in our mouth. We don't have them covering our bodies the way that a lot of fish do.
Because we're cowards.
There are still some interesting questions about sort of the origins of bone and how bone came about. For example, the exact relationship between all these different types of bones isn't fully understood. If, what is the exact relationship between dental tissue and bone tissue? Did one come from the other? Did they just both happen to evolve around the same time? Is the bony plates on the outside of the body of these early fish related to the internal skeleton, the sort of cartilage forming bone versus the membrane bone? Did one of those give rise to the other? Are they convergent developments of mineralization? Exactly the details of this diversity of bony tissues, of mineralized tissues and vertebrates, are not fully sorted out, which is very cool.
Yeah.
There is also the question of, when we look back at this time, we're looking at the Cambrian fossil record and we're saying, well, everyone's mineralizing and most of them are using calcium carbonate. So what was it about vertebrates that they turned to calcium phosphate? Why are our bones made of a different material than clamshells and trilobite exoskeletons and stuff? And this is one that we don't have a solid answer to this, although I did come across a couple of very intriguing hypotheses. One of which is that calcium and carbonate, CaCO, all of those materials that make up calcium carbonate, are readily available in ocean water, but phosphate isn't always readily available, right? Phosphorus is not. So it could be that this type of mineralization started in part as a storage unit.
Yeah.
It's like, well, we want to be able to hold on to phosphorus, so we're going to start building our bodies out of it. Another thing that I've seen pointed out is that phosphate is chemically more stable under certain circumstances and certain conditions compared to carbonate. Another thing I've seen pointed out is that phosphate is more chemically stable than carbonate in certain conditions, such as low pH. And one hypothesis points out that low pH is something that can happen in the body when it produces things like lactic acid, which is produced as a byproduct of high activity levels.
Yes.
So it could be that as vertebrates were more active, more mobile in their environment, their bodies would start breaking down calcium carbonate, and calcium phosphate might be more resilient to the kind of physiology you need to have if you're going to be swimming around and actively hunting things and stuff.
That is a really interesting point. Very cool. Yeah, because the first thought I had between the two was that the calcium carbonate that all of those different forms of sea life build their exoskeletons and shells with, they are pulling from the water
Mm-hmm.
the ingredients for it. Because that's the whole issue with ocean acidification, is that we're changing the available ratios of calcium and carbonate.
Mm-hmm.
So I was wondering if calcium phosphate was removed from that equation in a way that made it a useful thing. But it makes sense for it to be storage or better for our internal anatomy.
Yeah. From these humble beginnings, bone goes on to be arguably one of, I don't know, the top 5 evolutionary innovations in Earth history.
Oh, yeah.
Really important. All vertebrate diversity then comes from there. It also forms the backbone. It forms the core of our ability to study life through time.
Mm-hmm.
We're going to take a short break, and then after a brief musical interlude, we're going to come back and start talking about the ways that bone is important specifically for paleontology. While preparing for this episode, I came across a 2014 paper that had this quote in it referring to bones. It is this key apomorphy, that is a feature of vertebrates, which transforms the fossil record of vertebrates from the exceptional to the routine.
Yep.
Bones are the quintessential fossils. We have talked on this podcast a lot about different things that fossilize, different types of fossilization. Episode 137 was all about fossilization. Bones are the capital F fossils. If you ask a child to draw a fossil, they will draw you bones 9 times out of 10.
Yes.
So let's talk a little bit about fossilization. Hard parts. This is one of the core sort of principles of paleontology. It's one of the core bits of wisdom in paleontology. Hard parts fossilize better. The reason for this is that bones and teeth and shells are highly resistant to decay and weathering. They are not easy for scavengers to pick at or to eat. They break down very slowly because they are mineralized. This means that they tend to sit around in the environment longer than other tissues. When the skin and stuff has all decomposed away or been scavenged, the bones often are still laying there.
Well, it's like how after a fire or just a dilapidated house, the chimney will often still be standing.
Yes.
Because the house was made of wood, it was made of brick.
Yes. So the bones not only last longer, they last longer in good condition. And that means they are more likely to end up buried and preserved. They're also more likely to survive the process of being buried.
Yes.
Right? A softer tissue is liable to be destroyed simply by the process of being covered up by sand or whatever. Bones and teeth and shells, being resilient and hard, survive much longer. So it's not that they are necessarily better at being fossilized, but they are much more likely to be in the situation that lets them become a fossil.
Yeah, well, it's this is a weird comparison, but it's like if you're mailing stuff. Like if I'm mailing delicate things, a bunch more of them are more likely to get damaged in the process. If I'm mailing people bricks,
you're getting that brick.
The mailman will have to be unusually tough to ruin my packages.
This is why we don't attach messages to pillows and throw them through windows.
Exactly. Like, so if you just are more resilient, you're more likely to stumble into the situations where now you can be preserved.
Bone fossils tend to form in 2 main ways. There's lots of ways things can become fossils. The 2 sort of classic ways that bones become fossils are 1, permineralization. This is the process by which new minerals, so right, if your bone is sitting underground, groundwater is flowing through it, carrying all sorts of elements and ions and such, and those get deposited to form mineral. Permineralization happens when mineral gets deposited in the pore spaces inside of the bone.
Yes.
So the marrow, the cavities, even in some cases, the space between the cells gets filled up with new mineral. Any space left behind by soft tissues that decompose away, like where the marrow disappears, minerals can fill in, which means that a permineralized bone is a combo of original mineral, right? That original calcium phosphate mineral plus new minerals.
Yes.
The other major way that this happens is replacement, where the original mineral is changed or replaced. So this can be a change in the composition of it, right? Certain ions might get swapped out for new ions just as it chemically reacts with the sediment around it. This can result in things like calcite turning into pyrite by being replaced by pyrite, by gold type minerals, fool's gold, pyrite. Or it can be a change in the crystal structure, which is called recrystallization, where you can get calcite turning into aragonite, which are both calcium carbonate, but a different crystal structure, a different, slightly different composition that makes them different minerals. In this case, the original mineral of the bone has been changed into a different mineral.
Yeah.
It is not at all uncommon for a fossil to include both, for there to be a bit of permineralization, a bit of replacement, maybe a bit of recrystallization. This is very common in shells. It's very common in bones. It is exceptionally common in teeth because of how resistant and hard teeth are. This can also happen in cartilage, especially if it was calcified cartilage to begin with. We have fossil cartilage from fish and invertebrates and even dinosaurs. So bone and other connective tissues are extremely good at becoming fossils, and the fossilization process often preserves a lot of that original mineral structure of bone. All of this makes bones extraordinarily useful for paleontologists. Just to name a few ways, bones are the go-to place when it comes to vertebrate fossils, the go-to source for information on how to identify the fossil. Bone features, the anatomy of the bone, the shape of the bone, tend to be diagnostic to different groups. Because bones are evolving along with the anatomy of the rest of the body, a crocodilian bone looks like a crocodilian bone, and it looks different from turtle bones, and it looks different from dinosaur bones. There are features, just like if you see animals in the flesh, the skin of different groups of animals looks different. They have come by different evolutionary paths. Which bones and which features are the useful ones varies depending on the group and the context. Often it's skulls, because skulls tend to be very distinctive. With something like crocs or certain lizards, osteoderms, the skin bones can be useful for identification. I often remind people that for snakes, it's almost always vertebrae. Bones are also important for interpreting behavior and lifestyle of ancient animals based on the shape of the bones, the special anatomical features. The shape of the jaws can tell us how they were eating, and the shape of the limbs can tell us how they were moving. They are the core of biomechanical studies. Because bones are the anchors for muscles, there are signs on the bone of where muscles attached, and even the shape of those muscles. So by studying bones, we can interpret the shape and features of the musculature that attached to that bone. And some paleontologists will examine the physical properties of bone to examine things like how strong was it, how sturdy was it, how much could it resist twisting or bending, to get a sense of how were these animals moving with these bones. We can study how bone changes over the lifetime of an animal. These are ontogenetic shifts, episode 33. We can look at signs of injury or disease on bones, where an animal broke its bone and it later got healed, episode 84 for paleopathologies. We can study the internal organs using the bones that surrounded them. Right? The shape of the brain case can tell us what the shape of the brain was like.
Yeah.
Episode 121, the inner ear bones can tell us information about the sensory structures of these animals. Episode 178, the blood vessels and nerves that went through it passed through cavities and channels in the bone, so we can explore those features. Also, teeth. Episode 88. Go listen to that about teeth. And we can examine the chemical composition of bone, right? Isotope studies. Your bones are made of the materials you get from your surroundings. So the chemical composition of bone can reflect diet, and it can reflect habitat. It can also reflect certain aspects of how your body was functioning.
Mm-hmm.
All of these are ways that we can use a humble bone to learn a ton of detailed information about ancient organisms.
It's such a fascinating scenario because, as we discussed in the first section, bone is not just a static stabilizing element. It is a living part of the body, which means your life affects it and it affects your life. So we can learn a whole lot about the growth, the biology, the other factors of an organism's life through its bones. But also, because bones are the thing that tends to fossilize from vertebrates, it's the tool we've had. So we have pulled as much information from it as possible. So not only was it already a good source of information, we've gotten as creative as you could possibly get to get all of the potential hints from it that we can.
Yes. Well, and because bones are such a fundamental part of the vertebrate body, and because you have your bones your whole life,
yes,
they are just so core to the way that the body functions that they have a lot of clues in them. For more examples of the ways that paleontologists use bones to interpret the lives and behaviors and evolution of ancient animals, I refer you to the previous 209 episodes of this podcast.
Yep, pretty much.
But what I want to do now is zero in on a very specific way that we use bones, which is paleohistology.
Yeah, one of the prettiest ways also, which is cool.
It's a fascinating way to study bones. It is also a very cutting edge. It's a very modern way. Haha, yes, we'll get into why that was a pun in a second. Histology was also requested a number of times by the people who were listed at the beginning of this episode. So histology, broadly speaking, is the study of tissues at a microscopic level.
Mm-hmm, mm-hmm.
In modern science, this includes all tissues. Right, you can look at brain histology, you can look any tissue in the body. If you cut it thin and stick it under a microscope, you can examine the microscopic structure of that tissue. Paleohistology can also be used on any tissue if you get the tissue available, but most of the time we're doing it with bones, because bones, again, are typically what we get. And because, like I said, the mineralization process of fossilizing bone tends to preserve microscopic structure. Because if it's just minerals filling in pore space, then the original structure of the bone mineral isn't changing.
Yes.
And even if the mineral of the bone is being replaced, if it's being replaced 1-to-1, then the shape of those tissues isn't changing very much.
Yes.
So you still get the original shape of the bone, even internally, even if it has been fossilized and permineralized along the way. This means we can take a very close look at the microscopic structure of fossil bone to see the arrangement of the bone tissue, to see patterns of vascularization, to see where the bone cells were being. So we can see all of these microscopic structures of the bone, which, as I was saying in the first half of the episode, are reflective of the way that the bone forms and grows and the functions that it serves. The classic method for paleohistology is to get your bone and cut, this is why that was a pun before, and slice it into thin sections. A thin section is exactly what it sounds like. You slice like a little slice of cheese, and then that slice of bone is ground down with like a power sander or something, a grinder, to make it thin enough that light will show through it.
Yes.
And you stick it under a microscope and you look with the light shining through it and you can see the detail of all those microscopic features. I have gotten to hold and see some of these, thin sections of bone. I'm sure you have as well. The most recent time that I did was actually Davis at the museum, who was studying thin sections of gray fossil site alligator bones.
Yeah.
Which was very cool.
Yes, it's so good.
This is also the procedure used for studying petrology and rocks. You grind sections of in my geology classes as a college student, we did a lot of like looking through polarized light microscopes at thin sections of rock, and it's so cool.
Yeah, that's what I also was referring to. Once you get these thin window panes of fossil and rock, they're beautiful.
It's like stained glass.
It's so cool and pretty looking. You get all these different colors and these patterns that are just mesmerizing.
We'll have some pictures in the blog post. Now, this is a wonderful tool and technique. The biggest downside of this technique is that it is destructive.
Yes.
You have to damage the bone to do this. You have to take a chunk out of the bone. So not every place wants to. I remember at the museum, Sean was very, very vocal about like, let's not cut up that many of our bones.
Yep. Let's be careful about how many we choose to do.
These days, we can supplement. Now, obviously, there's still a benefit to doing it. We can learn a whole lot. These days, that method is supplemented with tomography, which is scanning of bones, using X-rays or CT scans to look at the internal structure of the bone basically, this is just using waves of energy to collect information about the shape. That's what an X-ray does. This is a great way to look at the inside structure of a bone without destroying the bone in the process.
Yes.
This can be done as virtual thin sections. So the way that an X-ray often works is you're getting a 2D slice of the internal structure of something. More and more these days, we are able to produce 3-dimensional scans.
Yes.
Of the interior of fossils and other structures, which is great because you can get much more detail. You get detail in all directions, which is very cool. With these techniques, we can examine the shape and structure of the tissues. We can see growth patterns, the way that the bone tissue formed. We can look at the shape of the canals and the cavities to examine cells and vasculature that was in there. We can sometimes identify remnant organic material, the remains of cells and proteins and stuff, which usually are done with staining. The way that this is, you'll get antibodies or certain chemicals that bond to certain materials. So you have your bonding chemical and you attach it to a dye, something colorful, and then you to put it very unscientifically, spill it all over your sample. And then anywhere that, if it's collagen or something, it's like, yes, we have come up with a material that will bind specifically to this protein, and we put a blue dye on it, and then we spill it all over our sample, and the blue dye now sticks to that protein. So you can image it.
Yes. It's like when you used to make arts and crafts as a kid and you would put the glue down on the paper and then cover the paper in glitter. And when you pour it off, the glitter only has stayed where the glue was.
Yes. These techniques have a whole variety of super cool applications and things that they can help us to learn about ancient animals. I will go through a handful I don't think any of these are things we haven't mentioned in other episodes. We'll go through a handful of some of the famous ones. Probably the most obvious one, the first one for me, certainly that comes to mind, and the one that I feel like has gotten the most written about it, is the ability to use histology to study growth in ancient animals.
Yeah, that's what I think of when I hear the word histology in a fossil study.
Yes. Well, it's those pictures of a leg bone that's been sliced in half, and you can see the growth rings.
Yes.
In the bone. That's what Davis was doing with the alligator bones at the Gray fossil site.
Yep.
That process, determining the age of an animal based on the bones, is called skeletochronology.
Nice.
Many animals grow in spurts. They grow inconsistently. So you will get faster growth during the good time of the year and then slower or no growth during, winter or the dry season or whatever season they're not getting enough energy to grow. And when they stop growing, it shows a line
Mm-hmm.
in the bone. We call this lines of arrested growth or lags, just like tree rings. It is
Yes.
almost exactly the same concept as tree rings, that more growth versus less growth at different times of the year.
And that's what creates those rings. If you could create a perfect scenario for a tree where it had ideal conditions nonstop all the time, you would get almost no noticeable tree rings because it would always be growing at the same rate. That change in growth is what causes them.
Yes. I think it might be true that certain environments you can't use tree rings. Allie's listening out there somewhere, and Allie can
Right?
tell me if I'm wrong. I want to say that like tropical environments, you can't date trees with tree rings because it isn't an annual process
Exactly. There's
of
other
their growth.
factors that are going to change their growth, not just the yearly season.
Same thing with animals. Not all animals this works with. Typically, we think of this with cold blooded animals,
Mm-hmm.
ectotherms, because they're growing in the summer and not in the winter. But you also get this in warmer bodied animals, sometimes just because they are growing at different rates at different times, or if they're going into hibernation of some kind. Anything that slows or stops their growth at a certain time of the year, if it's an annual cycle, now it gets one ring every year of its life. So
Yes.
you cut that bone in half and you can say, here, there are 7 rings here. This animal was 7 years old.
Yeah.
This is the most common way that we determine the age of, dinosaurs and other ancient animals. It isn't perfect. There are obviously, if something interrupts the growth of the animal at an unusual time, you'll have an irregular pattern. Also, sometimes the older lines will themselves be resorbed as
Yep.
the bone remodels itself. So you might not get the early years. So it's not perfect, but you can get a sense of the age of this animal.
You very often can usually at least say, at least it was this many years old. We
Yes.
might have lost some of the earliest rings. Some of these rings might be weird because there was a drought that messed up the pattern, but they definitely went through at least this many cycles that we can see clearly.
Yes.
Minimal age.
You can often also tell if it had stopped growing,
Yeah.
because as an animal gets to, most animals stop growing at a certain point. Even the ones that don't have like a set size that they're going to reach, eventually you reach an age where you just can't grow very much anymore.
Yes.
And as that happens, those lines become closer and closer together until there's just like a bunch. They're
Yeah.
bunched up along the outside of the bone. When you see that bunch up, the bunch up has a name. It's called an external fundamental system.
Ha.
That EFS is a sign of skeletal maturity.
Yeah.
This is an animal that reached its full size. So you'll hear us talk about studies, or you'll see a news about, oh, they found this dinosaur and it was still growing
Mm-hmm.
when it died, so it wasn't full size yet. This is often what they're talking about, right? Either something on the overall morphology of the bone, or in the histology, it had not yet reached full maturity. It had not developed those final structures that come when the bone isn't growing anymore.
Yeah. Which is a very cool concept that there is kind of a finish line that your
Mm-hmm.
bone reaches once you are as big as you're going to get.
Well, and this is different in different groups, right? In mammals, very famously, the way that many of our bones work is that they are unfused when we're born, and then they grow a bit, and then when they're done growing, the pieces fuse together.
Yes.
This happens to our skulls when we're very young. It happens to our leg bones. There
Mm-hmm.
are caps at the end of, your femur and your tibia and your humerus have bone caps on the end of them. And if you're a child, those bone caps are not fused on yet. When you reach adult size, they fuse together, and now your bone can't grow anymore.
Yeah, because now you can't just lengthen the middle shaft portion. You'd have to grow the entire thing as a unit, which is just too much.
So you can see these signs of, has this animal reached full size, in the gross morphology, right? The macroscopic morphology, and also in these lines that develop within the bone tissue. You can also get similar signals in things like teeth and scales and any tissue that grows regularly.
My favorite example of the fusion age determination is crocodilians fuse their backbones because the top and bottom of the vertebrae are separated, are two separate pieces, and
the
then
centrum and the lamina are two different parts.
They fuse from the tail toward the head.
Yeah.
So you can get a rough idea of how mature they are. So if you have a crocodilian with a partially fused skull, that is a fully mature individual. They are almost certainly at full size that they are going to reach, that they reached in their lifetime and would have reached. That is a extremely mature crocodilian.
Yeah. I want to say, I could be misremembering this, I'm pretty sure that elephants fuse from the bottom to the top.
Oh, right. Yeah, I think I have heard that.
That in elephant, like the vertebrae are the last thing to fuse.
So you can get these indications like a thermometer almost of
yes,
roughly how done are you in your process of maturing, which is very cool.
Looking at these growth tissues, researchers can also, there are cases where researchers have identified early life stress. So patterns of bone growth before that first line, that end of year one line, can indicate something about the health of that animal within its first year. If you can get enough examples, you can put together a whole growth curve for the lifetime of that species. One of the most famous examples of this was a study from several years ago with the dinosaur Maiasaura, where the researchers looked at the tibia from around 50 different individuals of all ages. And they put together, cool, here is how old Maiasaura got. Here is when they were able to say they reached sexual maturity at 3 years old and skeletal maturity at 8 years old. Here is how big they are at each age. We've talked about this with T-Rex is a very famous example where you may have heard us, especially in episode 120, refer to the fact that T-Rex had a teenage growth spurt just like we do.
Yeah.
This data is how we know that.
Yeah.
Is because you look at those lines across specimens of different ages and there is fast growth, wide lines during that growth spurt, and then they become closer together again.
And it's that lovely example of if you've got enough examples, if you can overlap the points they're at, now you get an entire life history for the youngest this animal could be and the oldest it can be, or at least the oldest specimens we have and the youngest specimens we have. And that's one of those things where that feels like a thing we shouldn't be able to know about an animal that lived 66 to 100 million years ago. Like,
and
that's
if they
so cool.
Weren't building their bodies out of rocks,
right?
We wouldn't.
Yes.
And this, typically we think of this with leg bones. This can also be done with other parts of the body. There have been studies that have used vertebrae to do the age studies with fish. Actually, Shay, who was on episode 198 with us, part of her thesis was looking at growth patterns in the gray fossil site fish vertebrae.
Yes.
I believe the vertebrae have also been used in snakes to do something very similar. Histology of jaws and teeth can be used to study rates of tooth development and tooth replacement. Right? How this is especially common in things like dinosaurs or I don't know if it's done in sharks, but they have that replacement where you can say, this dinosaur was replacing its teeth every 3 weeks or whatever.
Yeah, exactly. How long would it take them when they lost a tooth to grow a new one in place? We can get with this information.
You can also see evidence of age from things like bone loss, right? If you've got a whole growth curve, you can also say, all right, what starts happening to the bone when they get older?
Yes.
Do we see weakening of the bone? You can also look at evidence of bone remodeling. You can see where the bone has been resorbed. There have been some studies that will look at the histology and go, this area here between these two growth lines, the bone has been extensively resorbed, right? There's these pores, there's these cavities where the bone structure is different or less dense. That is the result of that resorption and remodeling process. And you might be able to say, yeah, this seems like it was probably a hard year
Yeah.
for this, right? It was running low on nutrients and had to start pulling from the bone. I came across a paper, this was just a few years ago, of a middle Ordovician fish, which is the oldest known fossil with signs of bone remodeling.
Neat.
That this is that process of bone. We can say how old that process is because you can see evidence of it in the bone itself. There
Very
are
cool.
also cases where you can get evidence of reproductive habits. This is another one we've talked about before on the podcast, the specific example of medullary bone. Medullary bone is a type of bone tissue that is formed today in birds, specifically birds that are about to lay eggs.
Mm-hmm.
A female birds will develop this medullary tissue, which is a tissue that just lines some of the internal spaces within their bones that functions as a store of calcium.
Yes.
They lay down, it's sort of like, putting together, collecting food for the winter. They lay down extra calcium for their eggshell. This kind of tissue has been identified in the histological analysis of several dinosaurs, including T-Rex, iguanodon, at least one species of iguanodontid, some ancient birds, ancient extinct groups of birds, also at least a couple times in pterosaurs, which is super cool because it tells us, A, these were females, one of the very few ways to identify the sex of an ancient animal. It tells us, B, they were of reproductive age, which is very cool. It tells us that they reproductively, their body functioned the same way that modern birds do, with sequestering material to then use for their eggs. And it tells us this animal was about to lay eggs.
Yes. It's very cool. It's cool that bones can do that. That we can use it as a store for these minerals and chemicals that we are going to need at specific times and hard times, but also that can be used as active storage, not just emergency storage, but preparative storage for a specific event, which is I'm about to make a whole bunch of calcium eggshells and it's going to take up a bunch of material. So I'm going to start building it up in my bones.
Yes, it's the equivalent of having a baby shower and everybody giving you diapers and stuff.
Yes.
It's like, I got to start stocking up now because I'm going to need these resources here pretty soon.
And the fact that we can find that in ancient animals, it's a very cool thing to be able to know at all. And as you just listed, tells us all these little details about that individual. That is very cool.
It's one of those things where every now and then I'll hear somebody make a comment where people will be skeptical of like, well, how can you tell so much detail about an ancient animal? And you'll hear someone make a comment like, oh, and it died on a Tuesday, blah, blah,
Yep.
Some sort of joke like that. And I often like to say, well, usually we're not making those sorts of super detailed interpretations. This is one of those cases where this animal died halfway through its 5th year of life. It had recently mated. It was about to lay eggs, but hadn't started developing the eggshells yet. It was a female. That's a crazy amount of stuff to be able to tell.
Yes, absolutely. Because so often for people, it feels like those scenes in Sherlock where he shakes someone's hand and tells them what they had for dinner last night. But every now and then we can kind of almost do that.
Yes. On a similar note, we're talking about sort of growth curves. Histology is often also employed to study rate of growth. So now I mentioned, you could get the spacing of those annual growth lines, but also the way that bone grows changes the structure of the tissue.
Yes.
In modern animals, right, birds and mammals tend to grow faster than a lot of reptiles and amphibians do. And faster growth in bone is associated with different arrangement of the tissues, more vasculature, more blood vessels and stuff. So there are cases, we've talked about some of these studies in the news and such. And this actually came up in episode 179 when we were talking about thermoregulation. There are a number of studies where researchers will compare the tissue types among different species to say, yeah, this species grew faster than this species.
Yes,
They
exactly.
exhibited, well, sometimes we'll refer to dinosaurs as this species exhibited bird-like growth, meaning it grew quickly, and we can see those features in the bone tissue. Faster growth is also often linked to higher metabolism or higher body temperature. And this gets really exciting, not only for telling things about individual animals, but whole evolutionary trends.
Yeah.
Studies like this on growth rate have been done on a lot of especially Permian and Triassic animals, the earliest reptiles, the earliest mammal ancestors, to get a sense of when did these growth rates go up. Who in the there was a study that came out not too long ago that did this with a bunch of Triassic reptiles and found, yeah, earliest dinosaurs were growing very fast. A
Yep.
bunch of these other reptiles were growing not quite as fast. Some of them were growing much more slowly. And you can get a sense of when in different lineages did higher metabolisms, faster growth. This is part of the reason why it is a common hypothesis these days that crocodiles may have lost their endothermy.
Yes.
That there's evidence that their deep ancestors were rapid growing, high metabolism animals, and that modern crocs may have reverted, just like those cartilaginous fish who went, yeah, we had this super cool feature and we don't even want it. And they
Yes.
moved back away from it.
And this is another one of those where it's so cool that one detail has to infer a bunch of other things, because you growing your bone faster, you can't just do it at the same density, because it was like if you're writing something, if I take my time and write one sentence, my handwriting can be perfect. If I have to write a paragraph in the same amount of time, I can't maintain the same perfect handwriting because I need to put down more words faster. So it's
This is
going to
why
change the structure.
if you ask a doctor, this is why doctors' handwriting is so bad.
Yeah.
Not because they don't know how to write properly, but just because they're writing all the time.
Yeah, and they're needing to get down as much information as you just said as quickly as possible because they have 5 more people they need to go see.
Yes.
And so like it changes the way the bone grows, but you can't just grow faster without having a different anatomy that allows you to. So like having that one little detail tells us a bunch of other things about the fundamental functioning of this organism.
Another thing that histology can be really useful for is understanding the relationships between not only different animals, but also different features of the body.
Mm-hmm, mm-hmm.
So just like, the features on the outside of a bone, when you just hold it in your hand and look at it, can be specific to different groups of life, so can microscopic features. You could get this type of tissue, this structure of bone tissue is only found in mammals or is only found in this lineage. So this can be helpful for trying to sort out affinities, especially in cases where the macroscopic features of the bone aren't doing it, like in turtles, where researchers have tried everything. And so there have absolutely been studies where they're like, well, maybe there's something about the microscopic structure of the bone tissue that will tell us where turtles belong. You can also look at the growth patterns and structure of the bone tissue to get a sense of how it formed. So I was talking earlier about how there are different ways that bone can form. Different parts of the body, bone develops in slightly different ways. So you can look at an unusual bone in an animal. This comes up with things like ankylosaurs with their plates and bony clubs on their tails. You can look at the histology of those and go, okay, these structures in the bone show that it grew in this pattern, which is what we see in bones like XYZ.
Yes,
Or you
exactly.
can look at the tail club and go, okay, based on the growth patterns, this club is actually 3 different bones
Hmm.
that grew individually and then fused together.
It's very similar when we look at like developmental tissue analysis and say, this part of the body developed from the same tissue that this other part of the body came from. So it is an extension of that thing.
Yes.
It's coming from the same source as they're going from a bundle of cells to a little baby. We can see where they come from. You can also do that with bone, looking at the structure of it.
Yes. This is also another thing that has been done with turtles.
Yeah.
Looking at their shells and going, what bone is this?
Yes.
What have you done?
Where did you get
Because
this?
where did you get this? Episode 60, go listen to us talk about how weird turtles are. One more category of thing we can learn from histology that I want to mention because it's super cool is, again, just like the features on the macroscopic scale of bones, certain features in the microscopic tissue of bone can be adaptations for certain lifestyles.
Yeah.
So, for example, I mentioned a number of times now that bone is the anchor for muscles. Well, the tissue of bone is structured differently at the muscle insertion points.
Hmm.
So, by examining the shape of that tissue, you can tell things about the shape of the muscle that attached there.
Oh, so it's not just the rough surface where the muscle grabbed on, but also the actual building of that section of bone.
Yes.
Cool.
You can also look at if there's cartilage preserved. You can look at how the cartilage was structured in and around the joints to get a sense of the mobility at those joints. The microstructure of bone can also inform things like stress and strain patterns. That came up in a very recent article that we talked about in the news about ankylosaur armor. I
Yeah.
believe that was a histological analysis where they looked at the microscopic tissue and they went, okay, here are the different layers of tissue. Here are the properties of each layer of tissue. And all together, here's how sturdy and strong this bone would have been.
Which is awesome.
Some really cool specific examples of this. There was a study, at least one study I saw referenced, looking at a group of early parareptiles, so near reptiles, procollophonids, which I think are Permian, where research noticed that their bones in cross section, the bone wall is extra thick, and the tissue where the muscle inserted was extra dense, which they interpreted as support for burrowing. That these bones,
Oh.
especially in the limbs, extra reinforced, extra strong muscle, which was supported by other aspects of their anatomy and where they were buried. Similar research has noted that the cancellous bone, right, the spongy bone, just the general pattern and shape of the spongy bone in ichthyosaurs and plesiosaurs is similar to the spongy bone pattern we see in whales and sea turtles.
Yeah.
That this internal bone structure seems to be an adaptation for open water animals.
Yes.
You also will get extra dense bone tissue in diving animals, where the density of the bone is helping them to regulate their diving. I saw one reference to a study on plesiosaurs. So these are the long necked, paddle flippered swimming reptiles of the Mesozoic. One study that estimated the size of red blood cells in
Oh.
plesiosaurs by measuring the smallest canals for blood vessels in the bones. And they estimated that plesiosaurs likely had larger red blood cells, which is associated with diving
Yeah.
because they hold on to more oxygen for deeper dives.
That's awesome.
How cool? How cool is that?
It makes complete sense.
Yeah, and there's been studies looking at the histology of like the air sac structures in ancient birds and dinosaurs. There's all sorts of super cool studies that have been done on here is a microscopic adaptation in the bone tissue for XYZ lifestyle.
Yeah. Very awesome.
This is one of those topics where when I was putting this whole episode together and all these notes together, I was like, oh, do we want, how much specifics do we want to go into? Do we want to do more on histology? And the reality is that all the other stuff that I have to say about histology is super cool examples of it.
Mm-hmm.
And again, go listen to every other episode of the podcast.
Yeah.
We have talked about super cool histological analyses in lots of the news. We've talked about lots of cool morphological studies and biomechanical studies. And if you don't know which episodes to go listen to for that stuff, don't worry, pick an episode. We almost certainly talked about some of this in that episode because, again, this is the core of paleontology.
Yeah, other than Alley episodes.
And even then we do news.
Yes, and in the news, you're almost certainly going to bump into something because, because a lot of the cool, exciting newses are usually some of like, hey, we looked inside the bone of this animal and found out that they were growing weird. It's like, that's a great news piece. That's awesome. So we usually talk about those.
Well, and it's one of the nice things, and even one of the missions of why we do the news on the podcast is because the main discussion tends to be more of an overview. And in our main episode discussions, we tend not to go deep into methodology or specific studies. That's the news. We get to do that. Every bit of news is here is the specific methods that they got to use.
Here's this cool new info and how they found it.
Yeah, so these examples, this topic of histology has come up over and over again on the podcast.
Yeah.
This was an extremely fun topic for me to get to dive into because bones are so cool.
They really are.
It's very cliche for me to have been geeking out about bones. Oh, I'm so excited to talk for 2 hours about bones. But yeah, no, they're awesome. There's so much cool information. There's so much cool biology within these seemingly very simple and fundamental tissues that we take for granted.
Well, and as has been evidenced by the fact that I think very likely more episode numbers have been said in this episode, the discussion, than the average.
Yes. This is one of the very rare times that I put episode numbers in my notes so that I would remember to mention. Oh yeah, Don't forget, mention this episode because, well, the number one recommendation for this one is go listen to episode 88, which is teeth, which is sort of the sister to this episode where a lot of these similar topics come up.
But like in so many episodes, we're discussing the bone, horns, we were discussing the bones, and tails is discussing the bones. And so we're constantly discussing them when we're talking about different habits. We're talking about them, but we've not gotten to really zoom in on what is this fundamental structure that so much of our discussion of biology, especially for us who are studied the bones of animals and not the soft tissue, what is it? What is this thing and why is it so unique?
Yes. Listeners, we hope that you have enjoyed this exploration of the bones that make up our bodies. If there's more that you want to learn about bones, check out the blog post, but also let us know what other topics you want to hear, and also go listen to other podcast episodes. Before we wrap everything up, there's one more thing to do, and that is our patron question. One of the benefits that you can get by being a subscriber on our Patreon at certain levels is you get to send in questions for us to answer here on the podcast. Will, what's our patron question?
Our question's from Ryan, who says, you've talked about isotope ratios as a paleothermometer, and specifically about how that has provided evidence for endothermy in prehistoric animals. Since the materials in bones gets replaced as fossilization occurs, how can we be sure the ratio reflects the temperature of the animal in life rather than the temperature the bones were exposed to during fossilization?
This is an excellent question and very related to the topic we've been discussing. How do we examine the specifics of bones? So the problem that Ryan has identified here is that, and this is true for isotope studies, it's true for any sort of microstructural study on bones, the bone is not the original bone.
Yes.
A lot of stuff has been lost. Some stuff has been replaced. So how do we know that the structure and the chemistry that we're seeing is reflective of the actual animal and not just something that happened during fossilization? This is an extremely important and common topic of concern with paleontologists. There are a few ways that paleontologists make efforts to get around this problem. One is these kinds of studies tend to target tissues and molecules that are most resilient, the
Yes.
ones that are least likely to be changed over time, like phosphates, or things like certain elements that are less common in the fossilization process. These are elements that are all over the place and they're just going to get into every bone, but this is a mineral that we only see natively in bone. It's
Yeah.
not coming from outside. Teeth are very often used for these sorts of studies because not only are they extremely resistant to weathering and chemical alteration, they're also more mineralized.
Mm-hmm.
There's less space, less organic material, less stuff to mess with the internal structure of the teeth. Paleontologists also have ways to check for alteration to the isotopes, to the structure. This is important. This is one of those things that we gloss over when we're
Mm-hmm.
talking about these isotope studies. It's the same thing as when we talk about like ancient DNA. We discuss it as though you just pull the DNA out of the bone and then you have it, as opposed to it being a dozen step process where you have to go through and clear contamination and double check this and all these precautionary steps to make sure that you're doing it right. When it comes to isotope studies and things like that, researchers first have to check that this bone has not been altered in a way that is going to mess up this analysis. So, a way that they might do that is to measure, in the case of isotopes, which are, the chemical elements making up the bone, they might compare isotope measurements from different parts of the same fossil. They might look at the teeth and some of the bones and some of the other tissues preserved, because if they've been altered, they should be altered the same way. Or I've seen some examples cited where researchers will compare different tissues in the same specimen. So
Yeah. Yeah.
they might look at the enamel and the dentine
Yes.
in a tooth and go, all right, this process of chemical alteration happens differently in enamel versus dentine.
Mm-hmm.
So
Mm-hmm.
if there's been alteration, we should see a difference in this composition between these tissues that wouldn't be there otherwise.
Exactly. Yes.
Researchers can also compare isotopes in the fossil with the sediment, and they might say, oh, well, this is something we wouldn't expect to be in this bone. It's all over the sediment, so that might be where it came from.
Mm-hmm.
You can also examine the physical structure of the crystals, of your apatite crystals or whatever mineral it is you're looking at, to see if they have been altered. If this all sounds complicated and like it would require you to know the specific chemical properties of any element that you might ever look at, yes, it does. Absolutely.
Yep.
There are different precautions you need to take for each different type of isotope that you want to study. It's the same thing with radiometric dating,
Mm-hmm.
right?
Yep.
We talked about in episode 167. If you want to do uranium lead dating, or if you want to do potassium argon dating, or if you want to do radiocarbon dating, each one of those has different steps you need to take to make sure you're getting accurate results before you can have confidence in them.
Yes. You can't just run them all through the same machine and it spits out the number you're looking for.
Yes.
They are each a different situation.
In the case of what Ryan was referring to, interpreting the body temperature of ancient animals, I looked up, I don't know if this was the specific study that we were talking about or that Ryan might be referring to, because there's been a number of studies doing this, but I did pull up one study example to see if there was anything else of note in there. And in that study, they were looking at oxygen isotopes in the phosphate of bones and teeth of early mammal relatives, because the way that oxygen breaks down into different isotopes is partly dependent on temperature.
Yes.
Not only did they do this with phosphate, which does not get altered very easily through fossilization, but in that study that I looked at, specifically, they are comparing isotope ratios between different species in the deposit. So they, at least in this case, and this has been the case, I believe, with a lot of other studies, they weren't saying this isotope ratio means this body temperature. They were saying these mammal cousins had a higher temperature than these reptiles.
Yes.
So it was
Yes.
more of a relative analysis than it was specifically what temperature are we looking at.
Yeah, exactly. We're not necessarily getting a number, but we're getting as far as the animals living here goes, they were the warmest inside.
Yes. So excellent question. Thank you for submitting that, Ryan. This comes up all the. I would get comments and questions from people like at the museum where someone would be like, "So when you're doing the dating on these fossils, do you worry about like contamination or like some sort of chemical alteration messing with it?" And I'm like, "Whoo, boy, do we!"
Yep.
Yes, like
Mm-hmm.
decades of research has gone into how do we make sure that when we're doing these sort of chemical or micro anatomical studies, we are not getting tripped up by whatever processes can alter these features.
Yes. Well, and it's why that you have people who are purely specialized in paleo dating and
Yes.
paleo isotopic studies.
Oh yeah. Or
Because,
histology.
yeah, exactly.
Yeah, Davis, again, shout out to Davis, who was in episode 13 1,000 years ago, who was doing thin sections of the gray fossil site alligator bones, learned how to do it by working with, I believe it was Holly Woodward, who is one of the top experts in paleohistology. This
Yes.
requires an expertise.
Yeah, there are technique. It's what makes those questions sometimes difficult when they're like, "How do you account for these things?" I don't, because
Yes.
I don't know how to do it.
I do not.
Just like, how does the statistics program work? I don't know.
Nope.
I'm not a statistician. I don't understand it, but I trust the people who did make it and who are specialized in dating these things.
Yes.
So I am going off of theirs.
I trust these researchers and their peer reviewers
Yes,
to
exactly.
know how that process works.
Yep.
So that I can be reasonably confident that these results are accurate.
But yeah, you get into these topics where it is so incredibly precise and delicate, even that not every paleontologist can just on a whim go, "Yeah, I'll do a isotopic study." Well, you're going to have to read some books first.
Yes. Speaking of reading some books, listeners, if you enjoyed these topics and would like to learn more about them, there will be links in the blog post. I mention that because one of the things that I referenced heavily for this episode and my notes in this episode is a book about paleohistology.
Heyo.
800 pages about paleohistology.
Just a quick read.
Yeah, just, when I was going to bed. Just real quick. Last night, I just quickly scanned it. Go check out the blog post. We will have pictures and images and more information, links to places, terms that we may have used in this episode. There's a blog post for every episode of the podcast. You can find all the episodes there. A big thanks to everyone who requested the topics that we went into in this episode. I'm so glad that we got to do an episode about bones, because it's just such a satisfying it just gets right to the fundamentals of paleontology.
It is nice when we get to because like typically we don't talk about bones this much, even if we get asked a question about bones. We don't actually get to dive into and it was really satisfying.
Big thanks to all of our patrons who support us here on the podcast, and extra special thanks to our top tier patrons, Jeff Ellington, Sarah May, and Danielle the Bug Lover. Don't forget that shortly before this episode came out, we just had our 8-year anniversary live stream. If you missed it, you can go check it out. It's archived on our YouTube page. If you watched that live stream, you would know that the first episode of our new series, Briks with Bask, is now available also on our YouTube channel. Go check it out. Also, go check out Gaming with Science, the podcast that we recently guest starred on, and played some board games. We release episodes every fortnight. Next episode is Darwin Day.
Yeah, it is.
It's gonna be a good one. Some sort of bone pun.
Yeah. It's the I was trying to think earlier of a joke that like this episode is like the opposite of the Bone Wars because it's just a peaceful celebration of bones and
Yeah,
not
like a bone holiday.
the Bone Treaty.
The Bone Festival. Thanks for joining us on All Bones Eve.
How many bones you need?
How many bones? How many bones you need? Welcome to Bone World.
Bonesville.
Bye, everybody.
Bye.
Thanks for listening to the Common Descent podcast. You can follow us on Facebook, Twitter, YouTube, and check our WordPress blog for pictures and links after each episode. Huge thanks to our patrons whose support helps keep this podcast running and who get access to bonus goodies on Patreon. The song you're hearing is called On the Origin of Species by Protodome, which we found at ocremix.org. Thanks again for listening. We hope you'll join us next time.